it's been a really interesting. I think it's been a really interesting journey over the last, say, maybe 5 years since AlphaFold has come out. Just seeing what is now possible with AI in the protein design space, and what kinds of problems this has unlocked, and really why we care about tackling them. So I'm going to tell the story about the journey from protein folding to binder design, and then eventually to antibody design, which is of course what our work has been focusing on. So as many of you may know, AlphaFold 2 really paved the way for generative protein design via protein folding. So the protein folding problem generally is, if you have a sequence of amino acids and you want to predict a biophysical structure of that, this, I guess, prior to AI is very difficult. People have tried deterministic models. It doesn't work super well. People have tried physics-based models. Maybe physics-based models work well for like local solving of structure, but don't really solve the global problem. And AlphaFold really was the first model to solve both of those things simultaneously. They won the Nobel Prize in Chemistry in 2024, along with David Baker for protein design. And I want to point out there's like kind of two separate problems that AlphaFold solves here. One is just the protein folding problem, but the other here is multimer protein folding. And I think like the way we think about multimer protein folding is it's actually quite different from individual protein folding. When you have like one protein structure, you can use a lot of evolutionary information to get maybe like global placement of that structure, and then do some like local understanding to build secondary structure and all those other features. Sometimes, depending on what you're folding in multimers, you can also use evolutionary information and then figure out where things dock. But we actually think AlphaFold is more powerful than that, and does some really interesting like physical refinement of docking of multiple proteins. So that's a lot of the basis for why we think mBER works, but we'll get into that in a moment. Okay, so once you can fold proteins, then you can ask some more interesting questions. So prior to this, we said, okay, given a protein sequence, can we get a protein structure? Now we can ask, well, what if we want to design a certain protein structure? Well, from a like synthesis perspective, from a laboratory perspective, you're sort of limited to working with sequences. You can design DNA, you can express those proteins. So we want to go from structure to sequence. That is the ultimate problem. But when we talk about like problems that are actually interesting to us from I guess from an industry perspective, or really just like solving problems, a lot of the time what we're interested in doing is not just making a protein that folds a certain way, but we want to make a protein that interacts in a specific way with other proteins in living systems. So this motivates the problem of protein binder design. So now we want to do a lot of things at once. We want to be able to fold maybe a known protein into a given structure. And then we also want to be able to design a new protein that will bind that structure. And in the last 3 or 4 years, there's been really significant progress in the protein binder design field. So notably, RF diffusion was, I would argue, like the first big protein binder design model, or even just like protein design model to come out. RF diffusion is based on diffusion models, which you may be familiar with from like image generation and more general AI problems. But yeah, RF diffusion uses diffusion models to generate a protein backbone structure. And then typically people will use a separate model called protein MPNM to then assign a sequence to that structure. So you're first designing the structure that you want to design a sequence for, and then given that structure, you're saying, okay, what's the sequence that folds to it? And oftentimes people will then fold that again without fold just to see if it's consistent with what you might predict it would look like. More recently, October of last year, 2024, Bindcraft was released. So Bindcraft is actually an entirely different paradigm for binder design. It solves the same problem, but rather than using a feed-forward diffusion model, Bindcraft actually does design the opposite way. So Bindcraft just says, well, if at the end of the day we're going to fold something with AlphaFold, and we're going to use AlphaFold to say like how good that structure is, why don't we just fold, like design something random, have AlphaFold fold it, ask AlphaFold, how good is that thing, and then directly optimize that through the AlphaFold network. So Bindcraft uses backpropagation through AlphaFold to design new sequences and new structures. Yeah, well, more on that in a bit. In terms of the impact of these models, both Minecraft and ArcticFusion have been shown to be experimentally successful. They're really the first experimental successes for protein binder design. But if you look at what comes out of these models, it is almost entirely what we like to call mini binders. So you can see some examples in the top right here in this animation. In this case, the red is the target and the blue are the binders. So you can see that the blue binders are really like just these alpha helical bundles. We think that like Minecraft and ArcticFusion rely very heavily on secondary structure to understand, what you can do with interactions. And specifically, like, there's not a lot of control you have in these models over what your binders look like. You can maybe bias them towards some different secondary structures, but you can't strongly constrain them. And that's a real problem in industry when we want to design things like antibodies, which are the sort of format of choice for drugs, for protein drugs, and are really highly constrained. Some quick motivation on the antibody design problem. It's maybe obvious, but if we just look at protein-based therapeutics approved by the FDA over the course of the last 40 years or so, increasingly the move has been towards antibodies, and actually just pretty much for all time, antibodies have been the majority class of protein therapeutics. So antibodies are really well clinically validated. If you come to a pharma company and you say, hey, I have an antibody that does XYZ, they'll be like, cool, we'll run through the motions to get that into IND enabling and so on and so forth. If you say I have this random mini binder that looks pretty weird and it just came out of my AI model, there might be a little bit more pushback to get that into the clinic. Although that might change soon. But anyway, we are really interested in designing antibodies. Big Pharma is definitely interested in designing antibodies. So that kind of motivates this challenge. And then this comes to our solution. So, mBER really is a combination of a bunch of existing protein design and understanding models. And we maybe use some clever tricks with AlphaFold, similar to what Minecraft does, combining it with protein language models, antibody-specific folding models, and our own internal wet lab feedback to successfully design antibodies that bind in the lab. So without further ado, I guess we'll get into how we actually went about developing mBER. And I also want to say, feel free to interrupt if anybody has questions. We'll also pause for questions at the end, but happy to take any questions in the middle as well. So the story for the development of mBER actually really follows the same timeline as the intro to the problem here. We were very early ArcticFusion users. I would say pretty much as soon as ArcticFusion came out, mostly Mike actually started working with it and designing binders that we were then testing in the lab. So by mid-2023, we had designed around 100,000 mini binders with ArcticFusion against more than 600 distinct targets. It might actually be over 1,000 targets. It's a big number. And we tested all those in vitro. So we were really interested in, are these going to work at all against our super diverse target set? But also, are there things that we can do to predict, which designs are going to work? In the original ArcticFusion paper, it's maybe not super clear how successful they are, so we want to do those measurements internally. This plot on the right is showing, this is specifically the IPTM metric from AlphaFold. So if you're not familiar with IPTM, basically if you, when you fold a multimer with AlphaFold, AlphaFold tries to predict how well positioned those two chains are with respect to each other. There's a number of metrics that you can take out of AlphaFold that sort of describe this prediction, but we found that IPTM is really strongly correlated with wet lab success. And you can see that in this plot here. So this blue line is showing the relative success rate for various IPTM ranges. And you can see that once we get into the sort of 0.7, 0.8 range, there's this big jump in success. So actually, if you can design ArcticFusion mini binders with that fold with IPTMs of say greater than 0.8, then you can hit 10% success rates in the lab is what we've found. That said, ArcticFusion does not often generate designs with that high IPTM. So the red line here is showing the underlying distribution of generations. There's a peak at the kind of low range, and then there's also things that are mid-high, but still maybe exhibiting like less than 5% success rates in the lab. Yeah, and again, just this pipeline is the typical RF diffusion design pipeline where we'll take a target structure, we'll use RF diffusion to design a binder structure, use protein MPNN to design the sequence, and AlphaFold to fold it and get these metrics. So we were using RF diffusion for a while. We were pretty excited by the results. But when Bindcraft came out, we were like, this is crazy. Because if you look at their success rates, they're showing that they are consistently generating binders with success rates basically ranging from like 25 to 100% in the wet lab. Here's a figure from their paper where they show like, for example, for PDL1, they tested just 9 designs in the lab, and 7 of them worked with at least reasonable affinities, although questionable in some cases. But the really interesting thing about Bindcraft to us was just the departure from the diffusion paradigm. So, if you want to like train a diffusion model, you have to be really careful about how you're processing your data. If you want to do like antibody design, it's especially hard because there just aren't that many antibody structures that are publicly available. So we kept asking ourselves this question, like how can we actually design antibodies if the public data is like maybe like 1,000 structures for VHH in particular. Well, Bindcraft shows that actually you don't need diffusion at all. You just use AlphaFold directly. And what we realized was that, like AlphaFold actually is capable of folding antibodies. Maybe it's not very good at it, but there's some things we can do to try and sort of trick AlphaFold into being better at folding antibodies. But just quickly, like why doesn't Bindcraft design antibodies as is? The way that Bindcraft works, it starts with a random sequence initialization. I mentioned earlier, like antibodies are highly constrained. A lot of this constraint is the sequences in what we call the framework regions. So there are a lot of residues in antibodies that are just pretty much always fixed, and you really don't want to change during your design process. Bindcraft natively does not allow for this. So pretty much the entire protein you're designing has to be brand new. Furthermore, Bindcraft actually completely does away with MSAs. So MSAs are multiple sequence alignments. They allow you to sort of sort out coevolutionary signal across proteins in multiple species. And we think that AlphaFold heavily relies on MSAs in order to position global structure. So this is a cartoon from Sergey Ovchinnikov, who's worked a lot with AlphaFold and actually developed the basis for Bindcraft as well. But here we're showing, or I guess Sergey's showing that in theory, if like this pair of residues co-evolves across a bunch of different species, then probably what that means is that those 2 residues are in contact in the structure. And AlphaFold will then use these MSAs to build up a contact map and get a really good global guess of the structure of the protein it's folding. But Bindcraft completely does away with MSAs. So Bindcraft basically is limited to binders that AlphaFold can understand without MSAs, and this is why we think that it largely generates just very heavy secondary structure conditioned binders. But yeah, so we didn't want to use MSAs either. MSAs are really annoying and expensive to compute. They're also not that useful for antibodies because the type of variation in antibodies is actually like it's not really evolutionarily relevant. There's like a prismatic mutation in a single individual. So. Essentially, we got past this by taking advantage of templating. So AlphaFold learns global structure through 2 methods. One is the MSAs, and the other one is just templates. So if you ask AlphaFold to fold something, there's a search against like all public structures that have already existed. And if there are like similar sequences in that structure data set, AlphaFold will sort of use that structural template as a guess for the global structure of what it's folding. And we said, okay, like what if we just like provide an antibody template and see what AlphaFold does? In the first case here, this panel A, we provide a sort of informative antibody template, this VHH template, but we keep the random sequence guessing. And here on the right, you can see actually the target is this sort of greenish protein in the back. The binder that we ended up designing is this mess in the front. It's the red indicates that AlphaFold's really not confident in this, and as you can see, it looks nothing like an antibody or even anything like a binder. We said, okay, well, obviously, like we're providing a random sequence. That's not going to work. What if we heavily constrain the sequence? What if we say, you're only allowed to design certain residues, but they have to be, sort of following the structure diversity of the variable regions of the antibodies. So on the right here, we're showing an example where we just provide that sequence information with no antibody template. You can see we get something that looks a bit more reasonable. Maybe it takes a trained eye to see this, but it's still pretty disordered. This is not a good binder. But when you provide both of these additions, both of these forms of bias, you can actually get AlphaFold to design and fold really high confidence and plausible looking antibodies. In this case, we're doing VHH, and a bit of background, I guess I didn't add in the slides here. If you're not familiar, VHH are just a single domain antibody. So it's a little bit smaller and a little bit simpler than full antibodies, but they're still antibody format. Here's some animations. These are just fun to look at. Basically, this is what the design process looks like in each of these three cases. So in the top left, you can see, again, like the sequence is random, and you're getting these like alpha helical bundles, but AlphaFold is maybe not super confident in for most of the time. On the right, you do get these nice beta sheet structures, but again, the confidence is very low. And in the bottom case, where we both constrain the sequence and provide templates, things are looking good, and AlphaFold converges on a nice binder. So we're claiming here that actually backprop is really all you need here. And we ended up doing away with a lot of the other innovations from Bindcraft. So one thing that's unique about mBER, I think, relative to almost every protein design method I've seen out there, is that we don't use protein MPNN at all, or any explicit sequence design step. We design the sequence entirely with AlphaFold. And that's to say we design the variable part of the sequence with AlphaFold. So after we provide this initial framework, we let AlphaFold do all of the CDR design, and we say we're going to trust you, and the binding interactions that you design, those are probably going to be real. We also want to solve the problem of, generating templates can be irritating, especially if you're using multiple different frameworks. We don't want to just grab a template from the PDB, especially if you want to fold, say, you want to fold a VHH template that's never been crystallized before. So to solve that, we simply drop in this model, Nanobody Builder 2, which has been shown to be a pretty good monomer VHH folding model. And for the sequence bias, again, the variable regions here have some structure diversity. We wanted to make sure we capture that in the bias, so we plugged in ESM2 to generate some of that sequence bias. So yeah, this is how we condition mBER for design. These are some brief in silico results. Let me get back to the backprop question in just a second. So just some brief in silico results. We're comparing this to the, maybe the state-of-the-art method at the time, ARF antibody. So ARF antibody is a diffusion model based on ARF diffusion, just fine-tuned on antibodies. Basically, you can see from the blue histogram here, most of the designs that you get from ARF antibody are very low confidence. Almost none of them actually pass your design filters, versus mBER generates high quality designs maybe roughly 20% of the time, which is a big improvement. Yeah, the backprop question, I'll briefly address it now, and maybe we can talk about it more at the end. The basic idea is, so normally when you're doing backprop, you have some loss function. So in this case, maybe you would say when you're training AlphaFold, for example, you predict a structure and you compare that structure to the known structure. And that distance between those two structures becomes your loss function. You can backpropagate that loss. You can take a derivative with respect to that loss and update the model weights to sort of improve the model over time, optimize the model to fold a better structure. Here, instead of optimizing the model, what we're doing is we're optimizing the inputs to the model. So we're saying, okay, AlphaFold is already trained. We can predict a structure, but also predict how confident AlphaFold is in that structure. And we say, well, if we just want to directly optimize that confidence, if we think higher confidence AlphaFold folds are going to be better, we just use the same backpropagation trick. We just update the sequence instead of the model itself. So hopefully that briefly answers that question, but we can come back to it more at the end if there's more.
Great. So we have mBER. We have reason to think that it's working in silico. And so now we want to put it through its paces. We want to see what we can do with it. And in Manifold, we're pretty ambitious. So we're really interested in pushing the limits as much as we can here. So we needed an experiment that can you know, help us achieve our lofty ambitions. And for us, that experiment is phage display. So if you go to the next slide, what we're doing here is we're taking bacteriophage and we are sort of genetically modifying them such that they display our designed binders at one end of the phage. So what happens, what this is it's a way of sort of very elegantly being able to control or connect a DNA sequence that gives rise to a protein sequence. So essentially that phage particle, it's a virus, and so inside of it is DNA that encodes for the protein that is now being exposed on the surface of that phage. And so what that means then is that if we can do an experiment where we can measure the function of, some measurement of the function of that exposed protein, we can then connect it through next generation sequencing back to the DNA, which then tells us what the protein was. So this is sort of a way of getting around the inability to just sequence proteins, right? Basically, by connecting the DNA, the nucleic acid that gave rise to that protein with the protein physically, by having that be a single atomic piece in our experiment, we can assess the activity of that protein in really, really high throughput. So what we can do is we can design a library of hundreds of thousands of unique binders. So we order the DNA in a big oligo pool of hundreds of thousands and make a library of phage where there are hundreds of thousands of different species of these phage that are all contain different DNA and thus presenting or displaying a different VHH on them. And what we can then do is, we can then take a plate of 96-well plate where we have different targets of interest arrayed on this plate. So in this example, we've got 24 targets per 96 well, so 4 replicates each. And we often do mini plates. And so we can wind up looking at hundreds of targets in a single experiment, where we're looking at hundreds of thousands of binders binding to those hundreds of targets. And maybe what's most interesting and most powerful is that we're not just evaluating the binders we designed to a single target, to that target, but we're evaluating the binders that we designed to target one against all of the targets. And same for target 2 and target 3 and target 4 and target N. So we have this sort of all versus all measurement of all our entire designed library against every target. And so that gives us both the sort of on-design binding rates, so we can detect whether the displayed VHH is binding to the target we intended it to, but we can also detect all of the off-design, whether the VHH is binding to targets that we never anticipated or didn't design it to. And we should say that all the experimental work wasn't done by Erik or I, it was done by our coworker and co-author, Supriya Ravichandran. So she's fantastic. So yeah, that's the experiment. But that actually creates another problem for us, which is that if we can do 600,000 VHHs, or even you'll see over 1 million VHHs and evaluate them, we have to design that many VHHs, which winds up being quite computationally difficult, actually, because you need a whole lot of compute power to do it. But again, we're very interested in going after many targets and really evaluating mBER as much as possible and creating as much diversity and interesting molecules that go down into our pipeline, our drug discovery pipeline. So we wound up selecting 436 human cell surface receptors. So essentially 436 molecules that are exposed on the surface of human cells and use those as our targets. Most of these don't even have a known crystal structure. We're just using the AlphaFold database as the template, as the crystal structure that we're designing against. So we sort of expect a high failure rate here, but we're very interested in the power of being able to design so many things and test them against so many targets is that we can sort of accept that many of them maybe won't work, but still go after the ones that do. So we're just showing you some plots here showing the number of binders against the number of targets. Our in silico results here are really interesting in that we see, maybe it's exactly what you'd expect. There's a high degree of dependence on the target for whether or not you're able to successfully design binders. So some targets, you get lots of good high IPTM, good confident binders, and some targets it's very, very difficult to design a high confident binder. So yeah, your mileage may vary for any given target. Maybe one of the advantages of not caring about any specific target and just going after 400 of them.
Yeah. Okay, we have a couple slides here for dramatic effect, and also just to show what some of the designs look like. Don't mind the secondary structure here; it's not super well folded, but these are real binders, and a lot of them do work. So we have you can imagine maybe designing to a bunch of different locations on maybe 8 targets. Of course, we did much more than 8 targets, and like I said, these are. Sorry, is there?
Yes, I have a question on the previous experiment.
Yeah.
I don't want to be left out. The experiment looks like if I'm right, the in vitro sort of lab experiment, and it looks efficient in ways of generating sort of antibodies that bind to the targets. My question or my intuition is, why was it done in the context of this study? I sort of like fell off why it was done. So if you could highlight me and in line with this work that is going on.
That's just a failure in our storytelling here. We will get to why it is done towards the end.
Okay. Okay.
But mostly the basic idea is just that you're very interested in a large number of targets and raising binders to a large number of targets. We'll get into what that's for in a minute.
A simple justification just for now is that if you want to know how good your protein design algorithm is, testing a few binders is maybe not enough to know exactly how good it is. You'd like to test as many binders against as many targets as possible to get that information. So there are deeper reasons, but for now, that's maybe a fair justification.
All right. Thank you.
All right. Anyway, we designed a lot of binders. There might be one more slide. Yeah, a lot of binders to a lot of targets, and also to a lot of epitopes on those targets. So I know these are really small, but you can see the binders in pink are jumping around to these targets. In some sense, we designed against much more than 400 targets because really every epitope on each target is effectively a new target. And we did this in a pretty naive way, and we'll get to that in just a few slides as well. But overall, what the experiment looks like then. So as we said, we designed against 436 of these targets. That ends up being a lot of plates. So we actually only tested against 145 of those targets. So here what you see, this funny stacked matrix. The way to interpret this is on the left axis here, every single row corresponds to a target against which we designed binders. And every single column corresponds to sort of that same target, but the hits that we called against it. And what that means is that the on-diagonal signal that you see here is binders that were designed against their target and also hit that target. The signal may look a little sparse, but actually it ends up being about half of all these targets, which is actually pretty much state of the art for the field. So the fun things about this matrix that you can also see though there are some weird single target effects. So you can see some vertical striations. What that means is that basically you have designs against targets that are not the one that's tested here, but that are still binding this target in the vertical column. There are a few reasons we think that's happening in this experiment. One is that some of the targets that we're expressing are actually just sticky. We did a combination of ordering targets commercially, but also expressing them in-house. Not all those targets are guaranteed to be well folded in vitro. And so if you have a sort of misfolded target, then you're going to end up having things sticking to it, even if they weren't necessarily designed to bind that target. Some targets also might just be naturally sticky charged, have a lot of hydrophobic residues, hydrophobic interfaces, things like that. But for the most part, you can see that this matrix is pleasantly sparse, I would say, in the sense that we really do want to see most of the signal on a diagonal, and that is in fact what we see. This is a pretty information-dense graphic. The other stuff here, just to be clear, so we designed, 1,153,241 binders. All of these were tested experimentally across 2 separate libraries. So that's why there's 2 matrices here. The targets that were tested in those experiments had some overlap, but there were also some targets that were mutually exclusive, which is, again, why these are not fully overlapping. And among those targets that were actually tested here, the number of designs that were actually on design is maybe it looks a little small. There's around 2,000 designs that they were designed against a specific target, and they actually were called as hits against that target. If we compare that, however, to the off-design hit rate, you can see that there's actually a pretty big difference here. So sort of on the per target basis, only 76 binders that were not designed against that target actually hit it. This whole thing raises the question of why are we hitting things that we didn't design against? One answer is, well, it's a really big library. And not only is it a really big library, but we're also testing against a bunch of targets. So when you look at other experiments in the field, a lot of the time what people do is they'll maybe design a handful of binders and they'll test them just against the target they were designed against. And that's useful to know if you're binding the target that you designed against, but you really have no guarantee that you're not also binding a slew of other targets that are in the proteome. So by testing against over 100 unique targets, we get this really rich off-design information, which tells us, are our designs polyreactive? Are they specifically binding the target of interest? And there's going to be some base rate of polyreactivity, probably no matter what your design strategy is, owing to just similarity in targets in the proteome.
Yeah, we really think specificity is something that a lot of the field underestimates or doesn't appreciate the extent to which a lot of these de novo designs and a lot of the de novo design success rates might be driven by sort of non-specific binding.
Yeah, and in fact, for some targets, like popular targets, and this isn't in the paper, but we tested, for example, against COVID RBD, which is a very popular design target. And what we found is actually that a lot of things just happen to bind COVID spike protein. It's just a really sticky protein. So it's maybe not surprising that you see pretty high design success rates against that in the literature. It's not even clear necessarily that those designs in the literature are specifically hitting that. They may just be sticking to it, which is, again, what we see for a couple of these design targets as well in our case. Okay, but if we look at this on a sort of per target basis, things actually do look quite a bit better in terms of hit rates. So the state of the art here in the field, around the time we were working on this, I guess we didn't include a lit review in this presentation, but some other groups that had claimed to do antibody design were like Nabla Bio. They were designing at rates of, I think they reported maybe 0.01% to 0.1%, maybe up to 1% success rates. RF antibody, again, was somewhere in the range of 0.1% to 1%. Recently, Chai-2 was released, but Chai-2 is, of course, a closed model. So they had maybe 10 to 50% hit rates. Basically, what we showed here is that for some of the targets, for the maybe best design targets, we do, in fact, hit those 10 to up to, I think, around 20, maybe 40% hit rates for those best targets. A lot of the targets still lie below that, but actually we're still close to the state of the art, if not exceeding it for those as well. So we exceed 1% rates for more than half of the targets that we hit in general, as long as you're filtering by high design confidence. And speaking of design confidence, so earlier we noted that in the RF diffusion designs, we saw there was a strong correlation between IPTM and design success. That remains true with mBER. Interestingly, it's actually maybe the hit rates are a little bit lower than what you might expect. So before we saw that the highest IPTM designs for RF diffusion maybe had 10% hit rates. Here in aggregate, we get closer to 4% hit rates, or maybe 3% depending on the library here. We think a lot of that has to do with targets. So if you were to subset only to the targets that had successes, those effective design rates do increase. But also there's a lot of challenges inherent with VHH. that are just not there when you're doing mini bindings. So I would say, even like a 4% success rate at this point is actually quite good for VHH designs. This is another view sort of presenting the same data, but looking at the individual targets that we hit. One thing that we're showing on this bar plot is you can see that as you filter by increasing IPTM cutoffs, your hit rates increase. And this is really nice to see. You might ask, like, how are you so sure that your on-design things are even like, doing what you expect them to do? Maybe you're like, just randomly hitting the things that you think you designed against, and if you sort it nicely, then it works out. That's not the case. When you filter by IPTM, you're saying, do I have higher success rates when I have higher confidence? And you see pretty much for all these targets that as we increase our IPTM cutoffs, and this is on a log scale, so these are really substantial increases, our success rates increase by, maybe up to even like tenfold in some of these cases. So that gives us extra confidence that our design structures are even like accurate. The fact that being more confident in your structure leads to higher experimental success sort of implies that those structures have some base accuracy to them. And yeah, if we look at the total set of 145 targets, again, maybe a 50% hit rate doesn't seem that good, but it is state of the art for the field. And yeah, we say, roughly 45% of these targets had statistically significant on-design hit rates relative to the off-design hit rates. Again, off-design hit rates being basically like anything that wasn't designed against that target. How often do we hit it relative to how often do we hit it when we actually specifically design against it? And on these plots as well, you see those off-design hit rates as the red line. So you can see that basically in all cases, our on-design hit rates drastically exceed the sort of random chance of design. So great for building confidence that we're actually
Erik,
doing successful design.
Erik and Michael, thank you. This is exciting. But on the previous slides where we had that matrix, I think you called it all against. Yes, where we.
Sorry, which slide?
The previous, yes, this one.
Okay.
My question is about the off design and on design.
Yes.
I think it was, yes. The on design, like when you look at the 76 and the 2200, I think, how do you normalize off design, like sort of hits, in a way to make a fair comparison between with the on design? I feel like that trends, the 2000 and the 76 needs some sort of re-explaining, if that's okay with you.
Sure, yeah. So the basic question for like what is an off-design hit rate and an on-design hit rate, the idea here is like what is the baseline VHH design for mBER? Well, one could imagine just like randomly sampling a bunch of VHHs and saying that's the baseline sort of hit rate. If you were like, let's say you made a naive library, you just had a bunch of random sequences and you panned them against some targets. Some of those would be hits, just due to the scale of the experiment. That's roughly what we're measuring in the off-design hit rates, but actually, I think our comparison is a little bit more fair, given that the mBER designs actually do have structured diversity. So we're not just doing random sequences, we're saying, what if I design against a different target? Well, now there's like still design information in that binder. That binder is expected to bind something. And then how likely is that binder to accidentally bind the wrong target? So for off-design hit rates, the question is, what is the rate at which a binder designed against any target that is not the one we're looking at actually hitting that target? For on-design hit rates, we're saying just binders designed against target of interest, what proportion of these hit? So did that clarify a bit?
Yeah.
One of the complexities here is that for a given design, it's being evaluated against one on-design target, right? But it's being also tested against over 100 off-design targets. So you can imagine, even if your on-design binding rate was 100 times higher than your off-design binding rate, which would be very good, it would tell you that you have a very specific design algorithm, you would still see as many on-design binders as off-design binders.
Oh,
right? You'd
perfect.
see just as much
Yes.
On design is off design.
Yeah, and in
So-
fact, in this earlier figure, we have this sort of star next to 76. This is a normalized number, so actually there are much more than 76 off design hits in the entire library. It's something like 100 times that. In fact, I think the normalization that we did exactly was just divide by the number of targets. But of course, yeah, the point is that just as Mike said, if your on-design hit rates are 100 times that, then these numbers are going to be equal, and it's not really a fair comparison. So that's why we've done that normalization.
Great, great.
All right. I think we can maybe move on to hotspot and epitope targeting.
Yeah, so next we just wanted to talk about one of the sort of methodological tricky bits about design and doing experiments at this scale, which is selecting hotspots. So if you go to the next slide here, we're interested in controlling where a binder binds on a target for a few different reasons. The first is that for a lot of therapeutic modalities, you might be really interested in avoiding or specifically blocking known epitopes, known binding interactions. So you might want to bind a target at a specific spot or avoid binding a target at a specific spot, or you might just want diversity, or you're trying to bind against many different epitopes because you don't know which one will have the sort of desired phenotypic effect. So mBER allows us to provide a hotspot where we can tell the model where to bind. This is also good diversification against, imagine all of your binders to a target, you try and bind to a single spot. Well, that spot might just not work, either because of maybe a post-translational modification or a disconnect between the sort of in silico model that you're designing against and the real structure that exists in vitro or in vivo. So by binding to many targets or many different epitopes on the same target, we're essentially subdividing it into many targets and thus diversifying our binders, hedging our risk that any given target or any given epitope doesn't work. But the third thing actually that's really important is that we actually think truncating, so hotspotting allows us to truncate the target, which we think is really important for a few reasons. So if we tell the model where to bind, we don't need to actually give it the entire context anymore. We can give it just a portion of the target and tell it, this is all you really need to know about, right? And that's really important for two reasons. One is the computational cost. These models scale n squared with the number of atoms in them, or number of residues, I guess. And so it's very expensive to design against the entire protein, and we need to design many, a million binders. So we can't do that. And the other thing is that we actually think that truncating the target actually helps the design model. When you have the full protein present, if you think about it, sort of like the problem is actually much larger. It's much harder for the model to understand where the given protein could bind, just because there are so many options, right, that it has to choose among. And so really restricting and constraining the problem space for AlphaFold, we think helps it arrive at better designs more quickly.
Yeah, we don't want AlphaFold to get distracted by unnecessary context.
Exactly.
Sorry.
So the way we do this is we need to make some set of chain breaks, right, within the structure, such that we can produce a truncation that ideally provides all of the necessary context while minimizing the number of chain breaks, while also avoiding any sort of bad context, misfolded things. AlphaFold put something there, not because it thinks it should be there, but because it has to go somewhere. But you don't want the model to see that and think that that epitope is blocked when it's really just an intrinsically disordered portion of the protein. And so for that, we used a dynamic programming approach. That's
yes, Michael.
Yeah.
About that very slide about hotspotting. I'm thinking now, when you're implementing hotspotting, like what are some of there would be like biological sort of priors that you are encoding in this process?
Yeah.
Yeah.
So the selection of hotspots, right? Like, how do we select what should the hotspot be, or are you asking sort of how we're actually telling the model what the hotspot is?
exactly like this sort of bias. I see like it's some sort of bias that is being introduced into the model to choose specific sites for binding. Is what I'm saying. So what is that information that is being encoded?
Yeah. There's two things. One, as Mike discussed, actually the truncation itself introduces that bias. So if you only give the model, the single epitope that you're looking to bind, then it's not going to design against anything except for that one epitope. On top of that, though, there is a hotspot loss term, which we actually took directly from Bindcraft. So essentially, it's just another one of these losses that we're backpropagating on, but we're saying, we're basically biasing AlphaFold to place the binder near a certain residue. So, in the sort of animated example here, that would be that residue in green, which is the central hotspot of each of these epitopes. And usually AlphaFold will behave and put the binder near that hotspot, but as you'll see in the next couple slides, sometimes it does not behave.
Okay.
Yeah, so we wanted to show a few examples of sort of different hotspots against different targets that we see. So this is the transferrin receptor. It's a fairly well-studied protein in specifically brain shuttling. And we're showing that we've got a few sort of low confidence binders to a few different epitopes on this protein. Another example here is FcRn, which is another well-studied protein in terms of its ability to prolong the half-life of antibodies. Here we're showing binding to a number of different epitopes. You see the successful epitopes, the successful hotspots highlighted in green, and the unsuccessful ones are highlighted in red, I should be clear. So each of these VHH structures in orange is a validated binder that worked in vitro that is now being shown here. So we, on this target, for whatever reason, we were very successful and we were able to design binders to a number of different hotspots on this target.
Yeah, I also want to point out with these figures, one of the interesting things is that so FcRn, we would say probably is like an easy target amongst our target set. But the way that we did hotspot selection, because we had so many targets, we actually randomly selected hotspots, more or less randomly. We selected from hotspots that were highly exposed on the surface of the proteins, but then we just designed against really all of them. And what you can see on the chart on the right here, part of it's covered up, but that's because there's no data here, is only 5, maybe 6 of these hotspot choices for this easy target actually resulted in successful designs. So even for easy targets, your choice of hotspot can really drastically impact your success rates. Another thing to think about here is actually what are the relative success rates? So on a per target basis, these circles were showing the success rates under different filters. So under the strictest IPTM filter, FcRn is like an easy target. You get 13-ish% success rates. But if you were to look at just the most successful hotspot for FcRn, it's actually substantially higher, around 30% success against that hotspot. So this kind of plays to the idea of different hotspots are actually different targets, different epitopes are different targets. And so even for an easy target, you might completely fail if you choose the wrong hotspot. And for a hard target, I would say maybe TFR is a hard target. And all these hotspots have maybe less success, but certainly these two have actually close to 10% success rates, given the scale of this. So yeah, even for hard targets, with good hotspot choices, you might be able to do quite well.
Yeah, and the last example here we have is PD-L1, just to show that one of the interesting things that happens here is that while we provide a hotspot and there's a loss term that drives the model towards binding that hotspot, it doesn't have to. So in the example of PD-L1 here, there's 2 hotspots that are on sort of different sides of the protein. And while we got successful designs to both of them, it turns out that even the successful designs to the hotspot on sort of the left here just created a binder on the good binding spot to the right. So funny, the models don't always behave super well.
Yeah, and this is part of why we think truncation is actually, in some ways, more powerful than the loss term that enforces hotspotting. If you truncate, there's no way for your model to design against the wrong hotspot. But if you just say, hey, can you please bind here, it might not work. And once again, PD-L1 is a relatively easy target, I think, for the field at this point. And you see, in fact, yes, so on the hotspots, we have up to 40% success rates,
That was
which is very good.
Erik, what you've just mentioned about truncation, I was thinking like when you truncate for long range, so you made a trade-off between losing that geometrical information and the computational efficiency that comes along and precision.
I would say the trade-off here is I would say you don't really lose. Ideally, you don't lose any information. You are just purely improving like the context that you're giving the model and your computational speed. So I almost think of it like imagine you're like prompting a language model and you want to have it solve a specific problem, but you give it like your entire like database of problems. Well, your language model is going to get like distracted. It doesn't need all that extra context. And it might not solve your problem as well. But if you can just cut to the context that matters for the binding, like the whole protein's not involved in the interaction, right? Like, stuff that's all the way on the other side of a big protein like TFR especially, maybe there's some like conformational dynamics here, but we're not modeling that in either case. And all AlphaFold really cares about is the interaction residues. So as long as you can keep those in the context, we think that truncation is just strictly optimal.
Sure.
Cool. All right, we have a little bit of time left. We want to make sure we answer that original question, which is, why the heck are we doing this? So I'll like take this away.
Sure. So we're really, at mNifold, we're really interested in trying to solve drug delivery to different tissues. So we think, there's a lot of medicines that have been shown to be efficacious, but that they don't have the right biodistribution, which leads to things like toxicity or just a lack of efficacy in the tissue that you need it. So we're trying to develop what we're calling the epitome, which is sort of a measurement of how binders to different targets behave in a living system, where they'll go. So, going back to the original set of 400 surface receptors in the human genome or human proteome, the idea is if we can make a binder to each of those and then evaluate how those binders behave in vivo, where they go in the body, then we can create this mapping and we can create this really powerful atlas that will let us design drugs that target to specific tissues. So if you have something that needs to go to the liver, you can attach a shuttle to it, or an importer, we call them, and deliver them to the liver or the brain or the kidney or the heart. So if you put it in that slide. So yeah, this is the dream, is that each of these different targets that we're going after is expressed in different tissues, and so by exploring this entire space, by using binders to everything, we can unlock the ability to deliver medicines everywhere.
Yeah, and just to let that sink in for a second, it's like we're really not just doing binder discovery; we're doing target discovery, right? It's like by being able to say yes, we can actually design against hundreds or even thousands of targets. We are no longer bottlenecked by saying like, oh, what if our one epitope or our one target of interest doesn't work? In solving the biodistribution problem, we can just say, what does every single target in the human proteome do? This can only be accomplished with the experiment that we actually don't talk about in the paper, which is our proprietary in vivo multiplexing technology. But briefly, we are able to also do experiments in vivo at the same scale as we do in vitro. So we actually take all the same binders that we generate from these libraries, and we put that whole library, that whole double library of over 1 million binders in a single living organism, and measure exactly where all those binders go to different tissues.
So this is an example of sort of what that data looks like. So this is data from a non-human primate, a macaque, where what we've done is we've taken all of these binders that we designed, put them into that animal, and then we're able to quantify the amount of each of these different proteins in each of the different tissues in that animal. So it's a single animal, and we can measure for each of these different binders whether they accumulate in the adipose, in the brain, in the heart, in the kidney. We're showing you here an example of 100 binders that were all designed to bind to a specific target. So a single target of interest. We're evaluating all 100 binders in the same experiment, but this is just a small sample of all of the binders that are in that experiment. So to sort of give you some more context, here we've got some anonymized 7 different targets of interest where we are screening screening them and measuring their uptake in all of these different tissues. And you can see that we have some interesting brain binders, so things that, proteins that when we put them into the animal, they go into the brain, as well as in the bottom one there's a very specific adipose shuttle that accumulates in the fat of the animal.
And the fun thing is that, like, when we design binders against these targets with mBER, even before evaluating them in vitro for certain targets, which we sort of know the in vivo uptakes, we can see that those design binders actually have that in vivo profile as well. And ultimately, this feeds into our entire drug discovery platform. So the idea here is we're designing proteins at scale, at million scale and beyond, and we're testing all these proteins in vivo, in vitro as well. And through this sort of screening, we can do a feedback loop and further improve our design so that in the next iteration, our in vivo results are even stronger, and eventually lead to a drug that we'll be putting in the clinic. All right, I want to just wrap things up here. Quickly say, mBER is available open source under MIT license. So, please feel free to play with it and design binders, and also feel free to reach out or open issues on the GitHub. The code is still in a somewhat nebulous state, so it's being worked on, but it's fully functional. And so yeah, please run it, and if you have any issues, feel free to reach out, and we'll be working on improving things. I also just want to make sure we get to say thank you to our whole team here. Obviously, this effort is not just me and Mike. We have a magnificent lab team. In fact, like something like 2/3 of the company is in the wet lab. Oh, you're good. Yeah. And also, special thanks to our co-authors, Supriya and Pierce. As we mentioned, Supriya did like all of the wet lab work for this, like pretty much single-handedly, which is incredibly impressive. And Pierce is our CTO and sort of directed the research. So yeah, thanks to them, and thanks to you all for having us to talk.
Thank you very much.
Take any questions.
Thank you very much, Michael and Erik. This has been a great show. For me, my final question is, I think about VHH. Like, it seems so attractive in this study, and I'm wondering, why is it so attractive for this sort of like de novo design? VHH. Why? What makes it so special?
Yeah, I think if you compare it to pretty much all the other antibody formats out there, VHH is the sort of smallest antibody fragment. And in general, it's maybe intuitive, like smaller things are easier to design. There's just less stuff to mess around with. VHH are like relatively stable. They don't have like weird like chain pairing dynamics like full antibodies. We think that it's also possible to design full antibodies with mBER. The same strategy, we have tried it and it should work. But VHH is just a lot easier experimentally due to the size. Yeah.
Yeah, it's harder to do phage display as well with larger things. Not impossible.
Thank you very much. I think that's all from me. I don't know whether the members around have further questions. If there is no, I want to thank Erik and Michael once again. Thank you very much. I was looking at the paper and I realized the GitHub link is there, and
yes.
I'll be looking at it immediately after this. Once again, thank you very much. I can't thank you enough, but thank you very much indeed. It has been a great paper. And
awesome.
Really
thanks so much for having us.
Yeah, thanks for having us.
Yes. So to the members here and to those outside the country, and to Erik, I want to say goodbye, and probably see you in our subsequent talks, our members. Bye-bye to Erik and Michael. Thank you very much today.
Take care.
Okay.